Halo, Assalamualaikum warahmatullahi wabarakatuh. Today is my first day to meet you guys in this 10 biology class, right? So, let me introduce myself. My name is Salsabila. You can call me Miss Salsa. From now, I am your new biology teacher. And I want to tell you that I live in Depok, especially in Sawangan, Depok. So you guys, if you are from Sawangan, Depok, you can come to my home to make a group discussion maybe. I will waiting for you. Okay, so now I wanna check your present. So if you are guys, if I tell your name, so please tell where are you come from. Okay, Abdullah. Okay, you are from Bogor. Oh. And then Aisyah. Oh, you are from Depok. Okay. And then Muhammad. Oh, you are from Parung. Okay, Parung. So this class is complete, right? Okay, before we start to study, let's pray together. Pray begin. Alhamdulillah. So now I want you guys to watching this video. Let's go check this out.
Coronaviruses are a large family of viruses, some of which infect humans. The coronavirus at the root of COVID-19 is the newest known member of this family. And like other coronaviruses that infect people, the new coronavirus causes respiratory disease, among other symptoms. At their core, coronaviruses contain a genetic blueprint called RNA, similar to DNA. The single-stranded RNA acts as a molecular message that enables production of proteins needed for other elements of the virus. Bound to this string of RNA are nucleoproteins, proteins that help give the virus its structure and enable it to replicate. Encapsulating the RNA genome is the viral envelope, which protects the virus when it is outside of a host cell. This outer envelope is made from a layer of lipids, a waxy barrier containing fat molecules. As well as protecting the precious genetic cargo, this layer anchors the different structural proteins needed by the virus to infect cells. Envelope proteins embedded in this layer aid the assembly of new virus particles once it has infected a cell. The bulbous projections seen on the outside of the coronavirus are spike proteins. This fringe of proteins gives the virus its crown or halo-like appearance under the microscope, from which the Latin name corona is derived. The spike proteins act as grappling hooks that allow the virus to latch onto host cells and crack them open for infection. Like all viruses, coronaviruses are unable to thrive and reproduce outside of a living host.
Okay, from that video, we know that today we're gonna learn about virus. As you know guys, virus nowadays is so viral and go international. But did you know guys what is the structure of virus, and then how can they are doing their reproduction maybe? Okay guys, before you know about how virus replication and then what is the characteristic and then blah blah blah, you must know that the study of virus is called virology. Okay, and first of all, you must know who has found the virus. Okay, first evidence of the existence of virus came from experiments with filters that had pores small enough to retain bacteria. In 1892, Dmitry Ivanovsky used one these filters to show that sap from a diseased tobacco plant remained infectious to healthy tobacco plants, despite having been filtered. In 1897, Martinus Beijering called the filtered infection substance a virus, and this discovery is considered to be the beginning of virology. So, tobacco mosaic virus is the first virus was found. Okay guys, did you know virus is living or non-living organism? Virologists consider virus to be simply infectious agents and not living. Why? It's because they are not cells, or we usually call it acellular, and then they require living hot cells in order to replicate themselves, and then they just have one nucleic acid, either DNA or RNA, never both. And then how the structure of virus? First, virus structure is rod shapes. It's like here. Viruses that are rod shaped have proteins packaged tightly into a helix. For example is tobacco mosaic virus. The next step of virus is round. Round viruses are composed of 20 identical proteins arranged into a spherical protein coat. Example is rhinovirus, which causes colds and flu. And the next structure is complex. Complex virus is comprise a large group of virus shapes that vary widely. The common shape is that of the bacteriophage, that infect bacterial cells. Like this. Okay, I want to tell you the basic structure of virus. And in this picture, there are bacteriophage structure and then influenza virus structure. You can see that there are protein coat, nucleic acid, and then DNA or RNA, and then membrane envelope, and then fibrous, and then tile. Okay, and then did you know guys, the virus has an economic importance sense. They have beneficial effects. Like virus can be vectors. Vectors is needed for genetic engineering of virus types of cells. And then it is hoped that bacteriophage virus may in the future be used to treat serious bacterial infection in humans, eventually replacing antibiotics. And then, did you know guys, virus has harmful effects too, and like a disease. Did you know guys, a virus can cause illness and disease? For example, the influenza is causes of rhinovirus, and then the AIDS causes of HIV virus, and then hepatitis is causes hepatovirus, and poliomyelitis is causes of poliovirus. But did you know guys that not just human who got the effect of virus, but also the animals and the plant. For example is tobacco plants can got the virus effect from the tobacco virus, and then the chicken will be got the harmful effect of virus like chickenpox, varicella zoster virus. So, how the virus do reproduction? Virus do reproduction from replication, and then there are two replication from virus. There are lytic replication and then lysogenic replication. If you want to know more, check it out.
This is the picture of the process of lysogenic cycle. In a lysogenic cycle, the phage genome also enters the cell through the attachment and penetration. A prime example of a phage with this type of life cycle is the lambda phage. During the lysogenic cycle, instead of killing the host, the page genome integrates into the bacterial chromosome and becomes part of the host. The integrated page genome is called a propage. A bacterial host with a propage is called a lysogen. The process in which a bacterium is infected by a temperate page is called lysogenic. It is typically of temperate pages to be latent or inactive within the cell. And let's go to the first step of lysogenic cycle. It is the page infects a cell, and then next the page DNA becomes incorporated into the host genome, and then in the next step the cell divides and propage DNA is passed on to the daughter cell. And the next step, under the stressful condition, the propage DNA is excised from the bacterial chromosomes and enter the lytic cycle. And then in the next step, page DNA replicates and page proteins are made. And then the next new page particles are assembled. And then the last step of lysogenic cycle is the cell lysis, releasing the newly made pages. In a lytic cycle, first is attachment. Influenza virus becomes attached to a target epithelial cell. And then next step, penetration. The cell engulfs the virus by endocytosis. And the third, uncoating. Viral contents are released. And then in the next step, biosynthesis. Viral RNA enters the nucleus where it is replicated by the viral RNA polymerase. And then the next step is assembly. New pages particles are assembled. And then the next step is release. New viral particles are made and released into the extracellular fluid. The cell, which is not killed in the process, continues to make new virus.
Okay, from our first meeting in this class, there are any question here? Okay, I think it's enough, right? And then the conclusion of this meeting is the virus is the infection agents, and then the first virus who was found is tobacco mosaic virus, and then virus has 4 structure. First is round, second is bacteriophage, and then the third is rod shape. And then virus can be factors to be the beneficial effects of human, and then virus can be a harmful effects of human, animals, and plants, like illness and disease. And the replication of virus is there are 2 replication. There are lytic replication and then lysogenic replication. So guys, I think enough for today. I hope you can learn again and again about virus in your house. So let's pray together to closing this meeting. Pray begin. Alhamdulillahirrahmanirrahim. Okay, I hope we can meet again in next week, okay? See you!